My name is Anthony. I'm a senior field application scientist with Sony. We're going to talk about the ID7000, it's our newest third generation spectral analyzer. We're going to talk about some basics, but also some more in-depth things about the instrument and how we're doing the things that we're claiming we can do. Okay. So it's in terms of an overview, I'm going to do a very brief, like conventional versus spectral. What's the difference in case some of you are familiar with spectral? I know some of you might be, but this will be quick. And then we're going to do an instrument overview of the ID7000 itself. We'll talk about standardization and unmixing and why standardization is so important and how that's sort of the key to how we're doing a lot of things that we're doing. And then we'll talk about what does that mean for workflow, data quality. And then I'll talk briefly at the end about our new spectral flow analysis software as well. I might have a couple other things at the end for you guys. Okay. So I'm sure most of you are familiar since you're sitting in this room. Conventional flow cytometers use long path and bandpass filters to sear and chop up the light before it gets to your detectors. Right. So if you have a system with 6 detectors, you can do 6 colors. Right. That's sort of how it works. I think everyone's on the same page with me on that. In spectral, we don't have any of those filters. Right. So conventional, again, you have your lasers, you have your colors, looks something like that. It gets all chopped up, right? In spectral, let's imagine that we have a cell, and that cell has 3 colors on it, right? So here's our 3 colors. And this is kind of what you'd see if you looked at any sort of spectral viewer, you see these 3 colors, they'd show them to you like this. But in reality, when a detector sees light, it really just sees the total light, right? It has no way of parsing out what the type of light it is, it just sees everything. And so really all that light sums, sort of like you see here. So in a conventional system, again, you have those long pass band pass filters. It sort of chops up the light. So anything not in those filters is lost. It goes away. The detectors only see what the band pass filters allow them to see. Okay. And then you can sort of quantitate it. You do your spillover, and then you do your compensation. To deal with that spillover, you subtract out the spillover, right, to sort of get the signal that it is that you're looking for. In spectral, there's no subtraction. And like I said earlier, there are no filters, at least on our system. Okay, so you're getting the total signal from all the colors and all the light all of the time. And what we do is we use many more detectors over our colors in the system. So we measure that light at many points, and we're basically able to generate a shape of the light. Right, so by using many detectors and seeing those different intensities, and then sort of drawing a vector, we get the shape of that total light. And this includes the autofluorescence as well, by the way. We'll talk a little bit more about later on. Okay. So where am I going with this? I'm going with this is we use what's called the weighted least squares method. Okay, so this is the unmixing algorithm that we use. You don't need to know any math for this. If you're anything like me, when you see the Greek letters, you run, right? Didn't get very far in math. All you really need to know is where the calculate button is. But what this is doing, in a nutshell, is it's taking your single color controls, it's looking at the shapes of them, okay, and it's looking at the shape of your total light, and it's sort of piecing it all together like a puzzle, and then it's able to unmix your different colors by their shapes instead of by their intensities. Right? Everyone with me so far? Does that all make sense? So that's sort of the general big overarching difference between spectral and conventional. On this system, obviously, you're going to still look at your data the way you're used to seeing it, right? You're going to see your dot plots and your histograms and your contour plots, whatever you want. So we don't need to, learn anything new there. And you can analyze the data in our software. You can export FCS files, take it to whatever your third-party software is of choice. But I think you'll find that there are a lot of really nice tools in our software that will allow you to do a lot of, nice things with analysis that you haven't been able to do before. So what are the big advantages of this? Specifically on the ID. All the detectors are on all the time. Okay, so you don't have to worry about like, did I turn on the right detector? Do I have the right filters? First of all, there are no filters, and you don't have to turn anything on and off. So whatever you put on the machine, you're getting everything from across all wavelengths. Again, that spectral data is unfiltered. Okay. Our single color controls, once you run them once, you can store them into the spectral reference library. They can be reused, they can be shared. It doesn't matter if it's a bead or cell, which we'll go more depth into. You can also remove or unmix autofluorescence, right? On a conventional system, if you have a really autofluorescent sample, the only thing you can do is try to find a brighter fluorochrome to outcompete that autofluorescence. That's kind of your only option. right? On a spectral system, you can actually identify the autofluorescence, treat it as a color, and mix it. And on our system, you can actually do multiple autofluorescence parameters, tools in the software to help you identify the autofluorescence. We'll talk more about that as well. And at the end of the day, you can run more simultaneous color. You can go bigger panel because you have much lower chroma options than you do on a lot of conventional systems. So what this means is you can get more consistent results with less frustration. It's sort of spectral flow. It's flow with no regrets, right? In a nutshell. And it's future-proof, right? Because you have all this detection range. There's no filters or anything in there. So even as new colors become available from all different manufacturers, you'll be able to run them. You're never going to be like restricted by filters and detectors and things like that. Okay. So a brief instrument overview. The ID really is designed for high performance. Got a really flexible optical design. It's configurable with up to 7 lasers. If you have all 7 lasers, you have 186 detectors in that instrument, which allows for really high sensitivity and resolution. The software workflows come with really advanced options. We've revamped this recently, so we have our new 2.0 standardization mode, which I'm going to talk more in depth about today, which really allows us to do some really awesome, powerful things in terms of not only good data, but also ease of use to get that data. Talk about what that means. And then we have the auto sampler. We're going to talk about the auto sampler is, I know a lot of you out there probably use a lot of auto sampling, plate loading, tube loading devices out there. This one's incredible. Okay. I've worked in flow for many years at this point, almost a decade, which is scary. I've used a lot of samplers out there. This one is all the things that we claim it can be and more. It's really, really amazing. So if you combine a streamlined workflow with high data quality, that's the Sony Spectral. Okay. So again, it's designed to support high dimensional analysis. We do a little X-ray here. We have our auto sampler, which by the way is included on every model. And it's not an add-on like you see on some other platforms. It's actually the instrument is built around the loader. So it's all one cohesive system. Right above the auto sampler is your flow cell. Then you have your up to 7 spatially separated lasers. And then each laser has its own dedicated PMT array. Right? And the only sort of filters, and it's not even really a filter, it's what we call a grading system. All that does is once the light gets to where the detection point is, it goes to this grading system to spread out the light so that it hits all the detectors. There's no really filtration of that light. And I'll show you what this means. And then on the left side is all the detection decks, one for each laser in the system. It is field upgradable, right? So you could start with like a lower 3-4 laser system and then upgrade it in the future to a 5, 6, or 7 laser system. Okay. So let's start with that auto loader. This is if you just open the door on the machine, this is what you're going to see. You have the option of running 24 tube racks for 5 ml tubes. There's a little sort of like 3-slot sort of system on the left side here where you can put your QC and your cleaning tubes. So you don't need to take up spaces in your plate or your rack for cleaning. It will clean from those tubes. Built-in thermoelectric cooling. So you don't need any water baths or anything. It will just regulate the temperature with a LTA cooling system. The washing block, which is going to wash the inside and outside of the probe after every single sample. And again, you can do plates on here. You know, UV, flat, doesn't matter. Mid-depth, deep well plates, 3D, 4 well plates with no hardware changes. So whatever you have, stick it on there, it runs. Okay. You don't have to give it any kind of like coordinates. You don't have to give it any kind of like dimensions of what you're loading on there. It just goes. I don't even know how to adjust it. I've never had to. Anything I've ever put on there for the most part works. It's really, really impressive. This is the agitation. So the agitation is not an orbital shaker and it's not a pipette up and down. The probe itself actually vibrates. So each sample is getting agitated at the time of acquisition. So your 96th sample hasn't been agitated 96 times by the time you get to it. And then that washing block comes down and washes the inside and the outside of the probe, again, after every single sample. So how does it do this? When the block comes down, it does an inner wash, right? So basically back flushes, then it vacuums that away to the waste. And then this other line over here pumps in fresh sheet fluid, okay, to clean the outside of the probe and then vacuums that away to the waste. By the time you move to the next sample, there is no fluid of any kind left over in that acquisition area. And so you essentially have 0 carryover. And I never like to claim 0 because that's a very bold statement, but it is really truly almost no carryover. typically claim less than 0.1%. All of these features are all configurable on a single well level. So you can run things to sample groups, you can run things to, sample by sample, configure different speeds and washing modes. We also have a low dead volume mode, which basically means you can pull everything from the well. And all of this, including it will monitor itself. So what that means is we have this event check feature. The event check feature will basically allow it to monitor the event rate over time. But if you have any sudden changes in event rate because of a clog or an air bubble, or you want a sample dry, it will detect it on its own. It will stop. It will deal with that issue. There's a little 2.5 minute back flush and cleaning to either get the air out of there or get the clog out of there or whatever it is, and then it will move to the next sample. Right? When it does that, it will also flag the well where that event is triggered. So when you come back from lunch or the next day or whatever, you'll know, oh, hey, these two wells had event check trigger, and then you can go back and either append data or data and time, do whatever you have to do to get rid of that issue. Okay. The low dead volume mode allows the system to basically, for each and every sample, each and every well, it will actually move up to the probe, it will touch the bottom of the well to know where that bottom is, and it will back off just a few millimeters in order to take up everything in that well. So if you're using like a U or a V bottom plate, it will basically take everything. Maybe it leaves like 1 microliter, but it pretty much takes everything. With a flat bottom plate, obviously you have that ring of fluid, so there's a little bit more. But if you're somebody who's like, I don't have a lot of sample, I want to make sure I get it all, low dead volume mode is your friend. Right? It'll take everything. So again, all of these features are configurable, sample flow rate, speed, offset times, all this can be configured however you'd like. There's also an automated shutdown. So what that means is you can program the automated shutdown. It will use the cleaning tubes on that little left side area. Those little 3 slots there. It will clean itself and it will shut itself off at the end of the day. If you choose, you don't have to do that. So a lot of our customers, once they have everything sort of set up and they have their templates and whatever, they come in, they put on their rack of tubes or they put on their plate and they say auto fire and they go home. Okay. Because it will monitor everything. And QC and all of that can be automated and walk away as well. So you come in the morning, turn on the system, it tells you exactly what to do, put the tubes in, you go, come back when it's finished. Okay. So again, it's up to 7 lasers. All these lasers are spatially separated. It is free space delivery, not fiber to the flow cell. So you're getting the maximum power of those lasers for excitation. That's sort of what that process looks like, right? And then once you have those signals from the flow cell, they are carried via fiber optics to the actual individual PMT arrays, where we sort of bounce it. It hits that grading system to spread out the light, and it hits all of your detectors. And this is happening for every single one of those detectors, for however many lasers you have. Okay, and each detector is a 32 PMT array. Okay, so that's where we get so many detectors from. Every single laser has a 32 PMT array. Okay, and these PMT arrays correspond to your ribbon plots. If you haven't seen a ribbon plot before, this is basically the raw data from every laser, from every detector off of each laser, and sort of gives you the total shape of what's happening in that sample. Okay, so basically those arrays coincide to the x-axis of each of those, and these are your individual detectors. They sort of plot against each other. So basically, what is this ribbon plot? Again, I just told you it's the raw data, but basically, if you were to look at all your cells going through the lasers here, here's a 5-laser system, you have all the cells going through, and you were to basically pause at a moment in time, right? So here's all your cells going through each of those detectors, sort of like histograms stacked on top of each other and turned sideways, right? If you were to pause this in a moment in time, you would be able to get a shape of whatever that cell is expressing, right? However many colors are in there. And if you do this for all the cells, thousands of times a second, like you do on flow cytometers, right? And you sort of see all these different cells, and then you plot them on top of each other, right? It would look something like this. And then you apply a density gradient to that. That's what the ribbon plot is. Okay? So it's basically being able to see all your data all at once. But again, you're going to be looking at your parametric data anyway. So you'll have this ribbon plot, but you don't have to pay too much attention to it if it's like scary or new, right? You're still going to have all of your dot plots and histograms and whatever else you choose to set up. And then with our new spectral flow analysis software, which we'll talk about in a couple moments later on in the presentation, you can take all this data, upload it to the cloud, and allow you to do dimensionality reductions, UMAP, flow sum, Disney's, things like that for high parameter analyses. It's all done with cloud, but you don't need like a supercomputer to do it. Okay. The other benefit of this type of detection is the ability to do close neighbor fluorochromes. So on a conventional machine, if you wanted to do something like Alexa 488 and GFP, you can't because they're in the same detector, right? And there'd be no way to separate them out. Here, because things like that actually do have separate, different shapes, they're close, but they are different, you can actually separate them out. So these are just some examples of some close neighbor fluorochromes that we're able to resolve on here. So like BB421, E450, BB421 and Pack Blue we do all the time. You know, APC and LX4647. Everyone always wants to do that one. That one we can do pretty easily. And this is all bead data, right? Bead data always looks kind of nice and clean. So here's some real data. Okay. So this is excitation from multiple lasers and detector array, obviously. But because those shapes are close but not the same, you can actually unmix them. Now that doesn't mean that the signals are not going to spread, right? You're trying to have spread because they're similar. So what I would recommend is to put them on mutually exclusive markers, right? So if you want to use APC on like 4647, maybe put one on 3 and one on 19, right? T cells, B cells. That way, if you have to gate on those double positives, there's no issues there, right? But basically, my take-home message here is they do unmix. So in all these combinations, BB515, Alexa 488, PE, Spark Yellow, Green 581, all these different combos. If you were to look at the actual single colors of these, I know this is kind of small, but they look like they're almost on top of each other. But there are differences there that you can utilize in the unmixing algorithm, and then unmix them. Okay. And some of it comes down to because you're looking at the total light, not one detector. Some of these colors have, you know, as you know, cross laser excitation, emission where you wouldn't expect, and you can actually use that to help improve unmixing. Okay. In fact, if you were to look at some of these on like a spillover spreading matrix, you could use that to help you design panels, but these numbers are like quite high in some of these cases where like you would never use them together on a conventional machine, but here it works okay. Okay. So speaking of using extra information to improve, one of the big advantages of the ID is the ability to have a 320 nanometer laser, deep UV laser. As of right now, this is the only platform on the market that has this laser. And the big take home, the big deal about this laser is you get an additional, right, 32 PMT array, which is cool. Well, really what that means is even if you are not using deep UV excitable dyes, you still get an advantage because any emission that's happening out there that you may or may not already know about is going to be used in the unmixing matrix to give you better resolution. And we find that in high parameter panels, it actually lowers the overall spread just by turning on that 320 laser, even if you're not using any of the like the Q dots or the star bright UVs or whatever is primarily excited out there. So for example, here's some plots where if you just turn it on and turn it off, you can see that that spread sort of tightens up and everything looks a little bit nicer. So have an example of a 41 and a 50 color panel. Okay, so here's like a spillover spread matrix of a 41 color panel. Total spread is 2695. You just sort of, you know, calculate it all up. But if you turn on that UV laser or deep UV laser, that 320 laser, it actually lowers to 2161, which is a good improvement, not massive, but it might make a difference in some of those, you know, rare events or whatever it is you're trying to detect. If you go even bigger and you go to a 50 color panel, here the total spread is 9661. As soon as you turn on that 320, it lowers to 7806. Okay, so the whole point of this is that even if you're not using deep UV excitable dyes, 320 laser can give you an advantage by giving you better resolution and improving your unmixing by just having that extra information. There's a lot of dyes out there now, like BB510 is a good example. There's a big emission from BB510 out off a 320 laser that like no one really knew about because no one was using 320 laser before, at least in this sort of application. So yeah, that's sort of the big deal with the 320, and it's an option on the ID. Spectral reference library is something I mentioned earlier. This is what's going to give you the ability to run your controls once, save them, and then not have to run them again if you don't want, and save you a lot of time and reagent and money. I want to put a little asterisk next to that for tandem dyes, but you have to be a little bit careful with tandem dyes, I just want to be honest with you. But people use the library with tandem dyes all the time and don't have a problem. You just have to be careful about how often are you running that tandem dye. Check for breakdown, right? Because as you all know, tandem dyes will break down right? And that synthetic bond, and the shape will change as that dye begins, or as that bond begins to break down. Okay, so what a lot of customers do is they'll sort of take their tandems and run them once, save them in the library, and then maybe like once a month, right, a couple weeks, they rerun it to sort of check the breakdown and see that they need to, deal with that. But the nice thing is you can actually mix and match. You can run some from the library, some fresh. It's a really, really flexible system. So the big take-home thing here is run all your controls, save them to the library, and then not have to deal with it, at least for a while. The controls can be loaded from the library before, during, or after your experiment setup and acquisition. So a lot of customers show up with their fully stained sample, run it, record it, get the data. Data's not going to change, right? And you have all your detectors all the time, so get the data. Life happens. You got dogs, you got kids, whatever. You got to get home. I don't want to have to run 40 controls. Great, get your data, do your controls tomorrow, do them next week. And it doesn't really matter, right? That library can be reused, it can be updated, it can be shared with other users. It can basically be done in lieu of running controls every single time, right? Like I said, you can mix and match controls from the library and freshly run controls. You can also use beads or cells. It doesn't matter on the system if you use beads or cells. I'm not going to tell you that we don't see small differences, but what I'm going to say is that in the end, it doesn't really matter because of the unmixing algorithm that we use and how we treat that data that we collect. Okay, I'll show you what I mean by that. Okay. So again, they're saved into the library and they can be applied to future experiments. And, which we'll talk more in depth about, it doesn't matter the voltage that you acquire those controls at. That is no longer important on the system, okay? So samples can be run at one voltage, controls can be run at another. I'll show you how and why that works. Okay. Once you have all of your data sort of acquired, you've got your controls, you've done your unmixing, there's one button that you can click that will give you an instant N by N of everything in your experiment. You can see what it all looks like, and if you have an issue, like you see here, this one's under unmixed. Maybe you have a mismatch because of a tandem breakdown. Maybe you have a contaminated control. Who knows what it is, right? But you can then go in and turn on a little spectral reference adjuster tool. It's a little switch that you flip, and then you can go in and just click and drag that and fix it, okay? And really what that's doing is it's making a small tweak to that one spectral reference, and then it's re-unmixing the whole thing from the ground up. So that when you then go back into your experiment, you will have multiple unmixing matrices with which to apply your data, and you can just choose one drop-down menu which one you want to apply. Right? So you have a really, really nice tool to sort of visualize what's happening with your data, correct any issues that you have, right? With great power comes great responsibility, right? So you don't want to just want to like throw in a bunch of stuff in the machine and manually fix it and be like, oh, good, publish. Like, think about why you have those issues, right? You should try to address those problems the next time you run it. You don't want to just be masking an issue. Maybe your little tube of antibodies is contaminated, right? And maybe you shouldn't continue to use that one. But you have the tools to fix it then and there for that analysis, and then maybe think about why this is happening. Okay? Question?
Oh, just a small thing. People often are afraid of using spectral cytometers because of tandem breakdown and such. They're like, oh no, it will be standardized.
Yeah.
I would say to that you have this absolutely the same problems of conventional cytometers. It's just conventionally have no way to fix it.
Agree. Yeah. I think you're absolutely right. On conventional machines, you have the same problems, but I think people sort of blind themselves to that because there are no options.
Yeah,
right.
If you can actually take the tape on the tandem and like notice that it's breaking down.
Yeah. So one of the things you can do with the library and the software is, let's say you have a tandem, so you have PDSci5, for example, and you run it a bunch of times over the last 3 months. If you save each of those entries into the library, you can actually, plot them over each other and see, and be like, oh yeah, this one is, you can see the shape shifting and be like, oh, this one's no good, and get rid of it, or do whatever you want to do. But you can actually see the issue, you can actually diagnose the issue, and now you can actually deal with the issue if you need to, but just look and drag it. But the other thing to keep in mind is, with the ability to mix and match, let's say you have a tandem dye that you know is beginning to break down or like not in great shape. Run that one fresh that day. Okay, don't pull it from the library. That way, whatever is in that tube is going to match whatever is in that control. The shape will be the same. It may no longer play as nice in your panel because things are spilling over a bit differently, but at least you won't have that unmixing issue of like you're trying to unmix with two shapes that aren't the same. Okay, so you have a lot of flexibility in how you do things on here. Right? So that brings me to, what are my options when it comes to controls? You have a lot of options, right? One, run them all fresh like you do now on conventional, which should be doing every single time. Option two is you've saved them previously. Import them, right? They're all there, or mix and match. Pull all your non-tandems from the library, run all your tandems fresh, however you want to do it. Maybe you show up and one of your controls doesn't look great. You're like, I don't really understand what's going on. Maybe I contaminated it, but you've got one that's bad. And you know that your lab mate last week ran the same color. I don't even care, I'm not conjugated for the same thing. Right, and your panel is CD3 and their panel is CD45, doesn't matter, right? 50 is 50 is 50. Right, so delete that one out of your experiment, pull that one in from the library, you're on this. Right, we do that all the time. Yes.
That's have you died from different company? And should you mention this is from Sony, this is from BD, like APC from BD, whatever. But last time, different company, different things may have little different.
So really good question. What I would recommend is any non-tandems are probably going to be the same from any company, for the most part. Like PE, APC, FITC, those are just, they're gonna be what they are, right? So those aren't the same. Tandems, however, you definitely should rerun them when you get them from a different manufacturer.
Oh.
And you definitely should rerun them if you get a new lot. Because that bond is synthetic, you can have lot to lot variations, you can have variations by manufacturer. So certainly anything that's a tandem and you get a new shipment in, rerun it, save it to your library. The nice thing about the library is you can save all this information in there as well. So you can save manufacturers, lot numbers. You can put custom expiration dates in there, so it will warn you when like you probably shouldn't use it anymore. All of this can sort of you can use the library as like your own chlorophyll database if you want. Right? Yeah, go ahead.
Since tandem dye it causes a lot of problem, why should we use it?
Good question also. Because if you're someone who's trying to do 30, 40, 50 colors, there are not 30, 40, 50 non-tandem dyes that exist. So at a certain point, you have to start using tandem dye because that's all that is available to us right now. Or maybe you're not doing that many colors, but maybe you have your panels designed in such a way where the best possible color choice is going to be a tandem because of where your other colors fall, right? So if you're trying to design a panel where you have like minimized spillover, you have all these colors stacked in different areas, maybe you do want to pick a tandem because that's going to be the best possible place in the spectrum. That's a great question. So everyone's always wants to get into the conversation of autofluorescence when it comes to spectral. It's a big hot button thing because we haven't really been able to do anything with it before spectral. So on the ID7000, we have what we call the autofluorescence finder. And this is something that's in the software. All you need is an unstained control of whatever the cell is that you are interested in finding autofluorescence for. Totally unstained. No viability dye, no GFP, like give it to me with nothing in it. And you can launch the autofluorescence finder, and this will allow you to basically laser line by laser line, identify where there is fluorescence, put a gate around it, it will show you the spectral references right then and there. And then it will basically, for each gate that you identify, it will treat each one of those gates as a separate new parameter in your experiment, basically. So each gate you identify is a new color. And so for each sample or each sample group, however you want to design it, you can do up to 8 autofluorescent parameters. Okay. So if you have whole blood and you want to look at autofluorescence of like lymphs, monos, and grands, red cells, or whatever, you can do that very easily. If maybe you don't have a really complex sample, but you have a really complex heterogeneous autofluorescence on a sample, right? Maybe you have like a lung aspirate, right? Or a liver cell, something like that. Those are very weirdly complex autofluorescent cells, okay? The autofluorescence finder can help you sort of parse out the autofluorescence. If you put one parameter on there, basically all it's going to do is take an average of all the light it sees and goes, okay, here it is. That's not going to deal with any of them effectively, because now you have one parameter to deal with it, and the cells are expressing in different ways. So if you can do it with multiple parameters, you get much better autofluorescence unmixing, right? I don't want to say extraction or removal because it's not going anywhere. You're just unmixing it as a color. Yes.
So if you can get the fluorescence pattern from a cell based on its size and fluorescence,
Yeah.
I guess you basically have a spectral signature for each cell even before it's stained. So
Yes.
That sounds like something you could do a lot with, like it's.
It is something you could do a lot
Yeah.
with. Yeah, so what I've seen a lot of people doing now who have, IDs and other spectral instruments, and something I've started to think about is. If I'm gonna be designing a new panel, or it's a cell type I haven't worked with before, and I'm concerned about autofluorescence, don't even think about the panel yet. Just take the unstained cells and run them and get the shape of the autofluorescence. Then okay, here's where my autofluorescence is, and if you're not doing a ton of colors, you can pick colors away from the autofluorescence, and then it's a non-issue. You don't even really have to unmix it because you're not interfering with it. But let's say, a lot of mammalian cells have autofluorescence in like the lower wavelengths, sort of like UV, violet, deep UV. And you have to use BV421, you have to use BV395, maybe your marker's only available on that. Great, unmix the autofluorescence from it. It works very nicely. And you don't even have to use the finder as well. By the way, this is like a tool that's really there for really complex samples. You can just simply go into your sample and draw a gate and be like, that's my give me the autofluorescence of those cells. And it'll just do it.
Yes. Do you have any experience with the live cell and the fixed cell?
Yes,
Do they change?
yes,
Does the autofluorescence change?
yes, it can. Fixatives will often add autofluorescence.
Fixative will
Fixative,
add more.
different fixative chemicals or whatever you're using to fix your cells can add autofluorescent properties. There's all different things that are done to, some treatments at autofluorescence, looking at treated versus untreated. We have a few customers, we actually have a paper on our website of a group out in Europe that was looking at fetal liver stroma. And they actually use the autofluorescence finder to basically say, okay, these different cell types actually all have different autofluorescent properties. And they were able to actually separate out the cell types just based on autofluorescence. I know drug manufacturers and drug developers are trying to look at this because a lot of drug treatments and cancer treatments are very autofluorescent. So do a treated and an untreated, and they can look at actually the autofluorescence, basically shape of their molecule, whatever their treatment molecule is, and use that in their analysis. So it's very, very, very helpful. And maybe it's not, maybe you're just working with regular normal lymphocytes, maybe you don't care, right? Because it's not autofluorescent, whatever, but the tools are there for you. Another question?
Well, it just sounds like it could be very useful.
Very.
You might not necessarily know what markers you need
Right.
to look at cell populations, but
Sure.
you can still see cell populations even without the markers.
Yes.
Because a lot of people, in research especially,
Yeah.
they don't work with something that they necessarily know, that's why they're
Right.
doing research.
Exactly, and because it's now just being treated as another parameter in your unmixing, the instrument just sees it as another color, right? You can actually make a plot and say show me autofluorescence 1 versus this, or autofluorescence 1 versus autofluorescence 2, and actually gate on that population.
Very interesting.
It's very cool. Okay, so even if you're not this isn't a big part of your analysis, it may improve your analysis. You may get better resolution because of, interactions where you may not expect. So here's an example of this is just the unmixing settings window in the software. So in this case, you have a 14 color panel. We've assigned one autofluorescent parameter here. So I just put add, gave me a parameter. I told it what cells I was interested in. So on my unstained sample, I just said these ones. I assigned it. The negative is always going to be zero reference because a negative of a negative doesn't make any sense. So it's just nothing. So I care about the positive, assign it, and it gives me the spectral reference once I calculate it. Right, so the black line is the autofluorescence, and I can tell right off the bat that my BV395 and my BV510 are going to interact with that autofluorescence because you can see the overlap of the shapes. Right? So then when I go into my data, I can see that for my CD11C, which is on BV395, and my HLA-DR, sorry, not HLA-DR, CD45RO, draw on those two respective colors. If I do with and without autofluorescence, I can actually see the resolution improve because that autofluorescence is being unmixed. Okay, so here the 11C looks much better. I can get all this information, and here the 45RO sort of separates out even better. Okay. The other cool thing too is once you've assigned this, if you notice there's a little checkbox over here that says use as AF. If you have multiple autofluorescent parameters, you're not sure if this even makes a difference and you should be caring about it, you can just check that box on and off. And as you do it, it instantly recalculates your data with and without autofluorescence. So you can actually see in real time how it's affecting your data, whether you need it or not. Right?
Anthony, you mentioned that there was the beats and cells, you don't need to assign these beats or cells.
No. No need to. Okay, so some of these things are a perfect segue. What I'm about to talk about, instrument standardization. Instrument standardization is a big part of our new 2.0 software update. And this allows us to do these things I'm talking about, like not care about beads versus cells. It allows me to not care about the voltages at what control and samples require that. It allows me to use the library with reckless abandon. Because I don't care about how things were required, what they're conjugated to, the carrier particle, the voltages they're required at. It's all irrelevant if everything's standardized. And I'll show you why. Okay, so when you run daily QC on this instrument, it's going to do a lot of things. I know you guys are used to running daily QC on everything that you've ever touched, but this is doing a lot. Okay, so this is going to do things like your standard laser delays and area scaling and all of that, but it's also going to calibrate the flow rate, right? So each and every day that you run daily QC, if you tell the instrument to run at 1, it's going to run at a specific flow rate. So if it's run 10, it's going to run at a specific flow rate. So you can always kind of know like how much fluid you're consuming, right? But the other important thing that it's going to do is it's going to do this spectral standardization and it's going to do this PMTV standardization, PMT voltage standardization. Okay, and what that means is, if I have a shape, right? In this case, we have a shape from the 637 laser. We run these align check beads, we call them. It's like our QC beads, right? The spectral signature of the beads is known, right? And they're manufactured to basically like fluoresce off of every laser array, so we can sort of see a signal and we can calibrate all this. That's compared to, but whatever you run this every day is compared to basically a master shape, a master log file where it knows this is what everything should look like. And if there are any differences because of natural day-to-day fluctuations on any other machine, each channel individually, not the whole array, but each individual detector is going to be adjusted slightly up and down to make sure that that shape is exactly where it should be. And this is done across all channels and all lasers to ensure that there's a very precise spectral output of each array, not only that day, but over time as well. So if I run a sample today and I run a sample tomorrow and all the settings are the same, I should get the same answer, right? Standardization you do, or very, 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 very close, right? So by having these PMT arrays standardized, right, we do this over a wide range of voltages, so that finds the maximum usable PMT voltage array for each, PMT voltage for each array. So no matter what voltage you choose to run your samples at, or your controls or whatever, the shape of that is not going to change. The intensity is going to change, right? All of this will move up and down. The shape will remain constant. Okay? So if I were to take this PMT voltage and boost it up, you can see that everything moves up. It's getting more intense because I'm turning up the voltage, but the shape has not changed. Okay? And that's what's important. So the same voltage should always yield the same spectral profile and intensity over time, right? To keep the voltage the same. Okay? Now keep in mind, this is a 10-fold difference, right? This is like 10 to the, what, 10 to the 5 to almost 10 to the 6. That's a 10x increase. But again, the shape is the same. So let's compare these two. We do a voltage at 4.25 and a voltage at 4.75. These are like these arbitrary, they're like a standardized voltage output that we use, right? We have an unstained bead in both cases. So we're gonna run the positive, and then I'm gonna run the negative of my unstained bead. Now notice on the 4.75, it is a little bit more intense, right? We're gonna see a bit more of the negative, but again, the shapes are all the same, okay? So what the system does is it removes the negative signal. No matter what control you run, bead, cell, whatever, when you run that negative, that signal from the negative is just subtracted out. Okay, so what you're left with is only the signal from the fluorochrome itself. Right? Doesn't matter the carrier, doesn't matter what it's conjugated to. That's the fluorochrome. Then we plot it on a linear scale and we normalize it to 1. And if you overlay those two, they are exactly the same. Okay, so the voltage doesn't matter. All right. And if you do this, if you're a math person, you do this mathematically, you plot all this out, the R squared is 0.999. Okay, very good. Okay, so again, if you do this in real time, it looks something like that. Basically, you can go up, up, up. But basically, the proof that the PMT array is standardized, the normalized fluorescence distributions, the line should be identical. even when they're acquired at different PMT array voltages. So here's all those measurements from 4 all the way up to 5. By the way, if you move from 4 to 5, a whole number, it's a log shift in emission or in expression, okay? So there you go, so they're all the way on top of each other, right? So not only is this nice, that day or today and tomorrow, but over a long period of time, this is important. Because if you run the same thing, in this case over 175 days, and you keep the voltage the same, in this case the standardized voltage is 5, it shouldn't change, and it doesn't. In fact, all of these points, the CV between one point and any other point is never more than 5%. Okay, so it's incredibly stable. To further bring this point home, if you were to take the same color, in this case Caravia Blue 520, which is a shameless plug for our exclusive Sony guy. But Caravia Blue 520, you acquire it over 4 months, right? This was 6 different voltage settings. Okay, so we just ran this 6 different times across many different time points, and then we put them all in the library and we overlaid them. Here's what it looks like. They're all the same. Okay. So changing the PMT array voltage does not change the distribution of the fluorescence. It only changes the measured intensity. The array voltage can be optimized for stone color controls and then re-optimized again for whatever your samples are, or vice versa. Doesn't matter the order you do it. Okay. The PMT voltages do not have to be the same. Okay, so in this new standardization mode, each system is optimized using the PMT performance of that instrument, which is also important. So even though everything is standardized, if you happen to receive an instrument that has slightly better performing PMTs, because every single PMT is different, like manufacturing, that's just sort of like a law of nature. The whole point of me saying this is that you're not capping anything. Okay, so you're not going to receive an instrument that has, really powerful PMT, but because you're running a standardization mode, we got to cap the power at a certain amount because blah, blah, blah. There's none of that. So you're going to have the full capability of your instrument. The detection is stable. The workflow is incredibly efficient because of these things, which I'll show you in a moment. When you run your samples, you're going to be able to get your maximum resolution every single time of whatever you put in that tube. I'll explain why that works in a moment. But your control and your sample voltages do not need to match, which we've seen. And you get precise and accurate unmixing because you can do it live, you can do it offline, you can re-unmix, you can adjust the unmixing. You're always going to get really nice unmixing. Okay. So when I'm talking about this new fancy workflow, right? What we've done is we took controls and we took a sample, ran the controls at a voltage of 4.78, right? And we ran the samples at the same voltage. Everything matches, just like you would normally do on any machine. Here's the data. Okay. So it looks pretty good, right? But you can see on your ribbon plot up at the top here, there's a lot of white space up there, right? So you could boost those voltages and get more resolution out of pretty much every array in there. But maybe you don't want to because, oh, well, I ran the controls, I don't care, it doesn't matter, right? Controls are done. Let's turn everything up, what we call 1 max. We just everything up synchronously, right? Bring all the arrays up at the same time. So I have this plot here highlighted because if you keep an eye on that, you can see the resolution improve. Right, so there are other plots where this happens, but I just want you to focus on one point just because it's easier instead of bombarding you with data. So you can see I brought everything up synchronously, so now we're up towards the top of the plot. Resolution looks really nice in a lot of these, better than where we had it before. But I'm going to take it one step further. Notice on like my 355, certainly in my red, I still have white space. I still could get more resolution out of those, but I can't because I boosted everything up synchronously. And I'm sort of at the top there. I don't want to saturate my detectors, right? Because it's not good. Go ahead and adjust those arrays individually. I don't care about the balance because when you unmix, because of all the standardization that's been done, the software is going to rebalance itself for you. So you do whatever you need to do to maximize those voltages, and it will unmix it properly. Okay? So I can then turn up only the 355 and only the red and leave the other ones alone, and my unmixing is still unaffected. But I'm maximizing my resolution in those 2 arrays. So whatever you got in that tube, from the voltage all the way up, right? And how do I know if I've gone too high? You can see the top of the ribbon plot there, right? So I would say maybe halfway between 10 to the 5 and 10 to the 6. But also when you're acquiring samples in your little acquisition dashboard there with your stop and start and all of that, there's a saturation readout in real time. It's gonna tell you in real time what percentage of your detectors are saturating. So you'll be able to know as you boost your voltages up as the sample's running, if you've gone too high, it'll tell you like 1%, 5%, 10%, it'll turn red, it'll give you like a warning basically. And then you can lower back down if you need to, so that you know that data is usable. Okay? And again, this was all done with controls at 4.78. Okay? And on beats. So you can go from everything the same, boost everything up synchronously, or adjust them all independently to get the best resolution, and it still unmixes properly. Okay, and that's due to all the standardization that we've done in this new software. Okay. So basically you have a really efficient workflow. You have a software that's going to work for you and with you and is very flexible. And this is because you have that library, also you're able to reduce reagents, you're able to reduce cost, reduce time, right? Your single stains can be on beads now instead of beads or cells or beads and cells or bring both or whatever. I like to do everything on beads. Obviously there's viability guys and things like that, of course, but for the most part, beads. Voltage does not have to match between samples and controls. You can reuse that library. Really, really, really flexible. So how does this all work? Right, this is sort of like one of those too good to be true kind of things. And I know I'm going to bring a lot of rules here. It all starts with the standardization, right? If the spectral shape from each array remains the same regardless of the PMT voltage, then adjust everything as you need to. If the controls are required to unbalance PMT voltage, the software will provide a coefficient that was generated during the daily QC to rebalance the signals when it's needed. Right? So if you don't adjust anything, or see if you go up, down, whatever, it goes, okay, that's fine. It takes the voltages that you've given it for your sample, and it applies the coefficients that it calculated from the daily QC, and it rebalances it for you and still unmixes it properly. The really cool thing is you can actually do all of this in real time as well. So if you already have a template that you've run and unmixing is already applied and all that, and you're just running a sample, it will show it to you unmixed in real time. So as the data is acquiring, you'll see it already unmixed. And if you're like, I want to turn the voltages up, down, tweak things, do that, and it will also unmix in real time as you're watching it, which is like really insane. But it does do it, which is cool. Okay. So again, those references are normalized. You're removing the raw intensity, leaving the spectral distribution intact. Okay. So basically, it calculates these coefficients, and it will be the same. Again, remember, the shapes are not going to change, so that the shapes will not change regardless of PMT voltage. The proper unmixing can be obtained by applying those voltage-based coefficients that were calculated during the daily QC, and the coefficients is determined by the voltage settings that you acquire your sample at. So it sort of knows what that base coefficient is, but it's able to adjust it based on the voltages that you choose. Okay. So let's talk about some special cases. Beads or cells, right? We're going to be having this argument until we're all dead, I'm sure. Okay. Your samples should always be brighter than your controls, right? Not true here. I don't care about the brightness of your controls. All I care about when you acquire those controls is that you can get me the shape. So all I care about is that your positive is separate from your negative. I want to see some separation, and I don't want it to be saturated. I don't want it to be so bright that you're off scale. Beyond that, I don't care about the intensity. Just I need to get that shape. And then that shape is going to be normalized, and then that normalized spectra is what's going to go into your mixing. It's also what's going to go into your library. You can reuse that library anytime you want. It doesn't matter who acquired it, when it was acquired, voltage, blah, whatever. Yes.
I guess, I assume it works the same as it works on our older Sony, that in the library you can see the shapes of things that have been already acquired.
Yes.
So then you can see my control looks the same as the shapes of 10 other people that acquired
Yeah.
GSP.
Hopefully.
Yeah, and if it doesn't, then there is a problem.
Then you have a problem.
Yeah,
Exactly.
yeah, so it helps a lot to see that everybody else got the same results, and you're like,
Right.
that guy,
Yeah.
that didn't have GSP.
Yeah, exactly. So just like you saw in those unmixing settings, it's going to show you all the shapes. It's going to do the same thing in your library. For everything that's in the library, you can filter it and say, only show me this, whatever. But yes, you'll be able to overlay things. You'll be able to sort of do all that. So yeah, you can compare. So you've got 4 Fitsies in the library, and then you run a Fitzy, you overlay them all, and yours doesn't look the same. You don't have Fitzy in it too, right? And you can be pretty sure of that. Okay. So here's beads on the right and cells on the left. Okay, we're doing the same thing just like we did with the voltages. Okay. We get the average intensity each detector. We do the same thing for the negative as well. You can see whatever these negatives are here have quite a bit of autofluorescence. Doesn't matter. That's going to be subtracted out. Okay. Plot it on a linear scale. Normalize it. And when you overlay them, they are the same. Okay? So the beads or cells argument on this platform, doesn't matter, whatever you want. If you want to run on cells, great, go for it. I think beads are easier, cleaner, right? I don't have to worry about like how much volume do I have, is the sample precious, whatever. I don't deal with any of that. Rare events, it's always a tough one, right? And again, 0.9992 if you're a math person. So, but what does this actually look like? Here's the same sample unmixed with a set of bead controls, and the same sample unmixed with a set of cell controls. And you can see that the unmixing is incredibly similar, if not the same. Okay, top, bottom, here's some other plots. All right, so you get very, very comparable results. So again, I prefer beads. I just think they're easier to use, tend to be less expensive. I don't have to worry about rare events and amount of volume and all of that. They have a consistent autofluorescence as well, right? If each sample that you're running is different autofluorescences, that can sometimes run into issues with brightness and things like that. Cells can have dim or rare populations, they already act like that. You guys know the story, right? So I prefer beads, but again, use whatever you'd like.
For the negative control needs to be your real sample.
The negative control being your real sample is only important if you care about autofluorescence.
Yes.
Right,
Yeah.
yes, but if you're running all of your controls with beads, then all you need is a negative bead. Or you can get on the internal negative if your control has that. Right? So what happens if the samples are brighter than the controls? Which you're never supposed to do, right? Big no-no. Okay? Here's compensation beads acquired at 4.5 with an MFI of 15,000 on that positive. Here's my fully stained cells acquired at 5.5, so I boost up the voltage. My MFI is now 398,000. Much brighter. Here's the unmixing. Okay, looks pretty good. And you might argue that like there might be some points where it's slightly over or under unmixed, but it's so slight. And for the most part, it's gonna look good, and remember, you can fix that if you need to. Okay. Here's another example where we use a dimmer control for a brighter unmixing, and we still got accurate results. So here's an MFI of 15,000, 25,000, 50,000. All the spectra would be equally appropriate for unmixing, because here they are in the library overlaid on top of each other, and they're all the same. Okay. So where do we get these spectral references? How are we doing that? We've sort of seen that already. But here's an example of BUV 615, where again, the spectral reference or the single color control or the spectral fingerprint or signature, whatever you want to call it. It's just the distribution of each fluorochrome's emission across the entire array, all the arrays, right? So here's the single positive, here's the negative. This is like real data. Okay, we look at the median intensity for both positive and negative. Then we subtract out the negative. Okay, take that, normalize it, and again, that's what's being used in the unmixing, that's being used in the library. Okay, so that's important. What if you have an extremely bright viability dye? Right, you've got one of those zombie or fixable or PI or whatever it is you're choosing to use. You don't have to sacrifice sensitivity to accommodate this bright viability dye signal. It's best to properly titrate the viability dye, so it's not the brightest signal, obviously. Let's say in that moment you get there, you're like, it's crazy bright, but I want to use the data, right? Saturate the viability dye signal if necessary, right? So when you're adjusting your voltages and you're fully stained, you're going to gate negatively on that anyway, right? For the most part, right? You're only going to care about the negative ones, the positive ones are going to be gone in your gating because those are you care about the live stuff. Saturate it. Who cares? Because if you have other fluorochromes on that laser that are important to you, you don't want to have those be very dim and not be able to get good resolution, right? So crank it up if you have to. Obviously, you go back and retitrate it and make it sort of more reasonable. But in that moment, you can crank it up and you're gating it up anyway, so who cares? Right? So that's an option.
Basically, this feature gonna help you. Doesn't matter your population is rare or the majority, and the expression of the protein is abundant or less. So you don't need to. Of course, it's better to think about use the dimmer color, the dye to stain your the most highly expressed one.
Yes.
The brighter one for the lower one.
Yeah.
That's the principle
Yeah.
you want to do. But you don't care. But this
Yeah.
picture will help you
It will, it's gonna be certainly more forgiving than a conventional system, but I would still say that all those panel design rules still apply.
Yeah.
Right, when you're acquiring the data, you can break a lot of rules. When you're designing a panel, it's still important that you follow a lot of those general rules, that hasn't really changed that much.
Okay.
Now, you can use different combinations than you could use before, but I would still advise you to put like the really bright markers on your lower expressors, right, and vice versa.
Yeah.
I would still advise that.
Yeah.
But it is gonna be a little bit more forgiving with that, yeah, for sure. A lot of people, I shouldn't say a lot of people, but I've seen it, many times where people have like a screaming bright GFP. Or TD tomato, right, because it's part of the cell line that they're using or whatever, can't get rid of it. My advice to you would be if you have a marker that is very important to your analysis, maybe it's a rare event. Or maybe it's just like, hey, my whole experiment hinges on this marker. Do not put that on the same laser where you have that screaming bright GFP. Because you're adjusting that entire array up and down, right? So if you've got screaming bright GFP and you go click, click, and you can't go any higher with your blue voltage, right? And your important marker is still way down here. There's not much I can do for you in standardization mode. Okay, so that's one example where this can kind of cut the other way. Where if that's important to you, I would move that to a different laser. That way you can say, okay, I can lower my signal on my blue to get my GFP on scale, but I can still crank up my other laser to get that important marker on scale. So think about that, because you're not adjusting detectors individually, you're adjusting arrays. And those arrays consist of like 35 detectors. Okay, so you may want to think about that a little bit. Okay. But again, you can still lower the PMP voltage if needed, right, to accommodate that bright signal. Right, you don't always have to go up, you can go down if you need to. So what is the conclusion in all of this? The conclusion is all of this is that in general, we see that the ID 7000 offers superior data. There are many instances where someone can show me a panel from a different machine where on one given day it looks better than the ID data for whatever reason. Maybe the voltages weren't high enough, maybe that panel was designed for that instrument and it's taking advantage of some things. It's very, very difficult to design a panel that looks beautiful on every instrument that you put it on, right? Because every instrument has little differences here and there. But overall, we see that the ID data looks very nice compared to other spectral instruments. So this is just an example of some of that data where you'll see really nice resolution and separation compared to competitor instruments. Okay, so you can see some examples here and here as well. That resolution looks pretty nice. Better separation. Okay. So the take-home message is that you have a lot of features on the ID that are going to make your life a lot easier. Automated QC workflows, there's experiment design wizards, so when you're setting up all those controls for the first time, it'll show you like where to put them in the plate and set up all the worksheets for you and the positive and negative gates. You don't have to build any of that. You have the library, simplify your panel design, right? Think about it. So if you have things in your library and you're kind of like, well, what would it look like if I use this control instead of that control? You don't have to run the whole experiment again, just swap them. Right, and to like re-unmix it. You can re-unmix things as many times as you want. Alright. You have that autofluorescence finder, the ability to do up to 8 autofluorescence parameters. On mixing flexibility, do it live, do it, in the moment, do it later on, do it tomorrow, doesn't matter. The auto sampler, right? You have active agitation, cooling, loaded volume, uncheckable carryover, cleaning, all that's already built in for you. And then the system will also monitor itself and shut itself down if you choose. Those are sort of all the big take-home messages of the ID. Question?
Well, kind of a comment also, so like our Sony Spectral has a number of advantages, which I suspect A70000 already has, just from a user perspective.
Yeah.
So one big advantage compared to other instruments is that with the SP6800, you can probably go from cold to getting some data in about 5 minutes, to getting good data when the laser's fully warmed up, maybe 10. So most other instruments are like 20 minutes to half an hour.
Yeah.
Especially considering that you can also use controls in the library, whereas most instruments you can't.
Yeah. There's pretty much, I don't remember exactly the specs on the SP, but I know on like all the modern Sony instruments, there's pretty much no warm-up time for the lasers. Right? If you can like turn on the machine and run it, and it'll be like pretty good. On the average day on the ID, from like the machine is cold, sat overnight for running samples is about 20 minutes. And that's without any laser warm-up. It's gonna do a priming, and it's gonna have you run QC and all of that, but that takes about 20 minutes. And you don't need to be there for that 20 minutes. Right? You can come on, you basically hit the power button, go to the software, it goes like, you should prime. And you hit start, and then it just does it. Right?
Yeah.
So that's the startup. And there's other things that you can do along the way. One really cool feature that I think we don't talk about enough on the ID, is the optics alignment. It will align the lasers by itself. Okay, it will remind you to do this once a week, a little wizard that pops up and goes like, it's been a week, you should do an optics alignment. Uses the same beads we use for QC. It's done in 5 to 10 minutes, okay, so you no longer have to like call the service engineer, have them come in, take it apart, get the Allen wrenches, tweak it, this does it on its own. Okay, so in terms of like downtime in a core environment, that's a really nice feature. Anytime it is not actively running sample, it just idles itself. Just by default, so like it shutters the lasers, there's no fluid being consumed, it runs on water, by the way. You don't even have to use sheet fluid, so there's no salt to worry about, okay? So from an ease of use perspective, I don't know anything easier, truly. There isn't really any maintenance for you as the owner or as the user, there's no long clean. There's no like, we're not gonna give you like a spare parts bag. It just goes. Right, yes.
An additional advantage we can probably expand on is clogging. We
Yeah.
haven't really seen much clogging on the SP6800, it appears to be almost impossible. Now it does get dirty.
It's a bold statement, yeah.
Well, it is.
Yeah, no, it's
like,
true, I
like,
used to run an anti-love with great machine.
Yeah,
Yeah. Yeah,
well, okay, bacteria once, but it was the LA.
yeah, it is, here's what I'll say about the ID, I don't wanna sit there and tell you it's impossible to clog, because it's not, I've seen it clog. But it is way more difficult to clog it than other instruments, we've taken tissue samples out of a freezer, thawed them and ran them, and it did not clog. Okay, so like it knows it very well, it has a very short sample path, it's maybe like that long from the auto loader to the flow cell, it's like 2 or 3 inches maybe. So you have a lot less highway for things to get clogged in, it's also doing that cleaning like constantly pretty much. Here's what I will say. When I have seen it clog, I have never seen it not recover from that clog on its own. So every time I've seen it clog, it has dealt with it and has continued running all by itself, I have not had to do anything. Right, so that's very nice, especially if you are not at the machine when that happens.
Do you think that the extra UV laser make a huge difference?
I think it can, we've seen some really nice data come out of using the 320 laser, we have quite a few users that have it and they love it. I don't think it's integral to getting great data out of the instrument, like a 5 laser system works beautifully, right, you'll get really nice data out of a 5 laser system. All that data that I showed you with the overlapping colors and all that, that was from a 5 laser system, the 320 wasn't available yet. But I think if you're interested in like autofluorescence and different types of funky stuff, or you have people that you know are trying to push for really high parameter panels, I would go with a 320, I think it's a big help.
5 laser can copy.
We've done 40 plus on 5 lasers, like we did 42 color on 5 lasers, I think we pushed it even higher on 5 lasers. So certainly, I mean that 50 color panel that you saw. We didn't see the data, but you saw that cellular spread matrix, we ran that on both a 5 laser and the 320. Just toggle it on and off. So I don't have like the data to show you, but it is certainly doable on 5 laser system. Yeah.
Yes, one last comment on the Sony software.
Yeah,
Is that it's not something that you can easily show in a presentation, but it is a big difference.
it's a big difference, it's very easy to use, it's very intuitive, it's different. Right, it's a little bit different than Viva or what, you know, Spectral Flow, whatever it is you use now. It's more
like a
intuitive,
silly company.
yeah, it's one of those things where. I know everyone's afraid of change, right, that things are different, I know this, I'm comfortable with this. All the customers that I train on the ID, I find that once they sit with the software for like 30 minutes, they're kind of like, I love it. It's great, it's intuitive, all the tools are amazing. Here's the other thing about the software, the base software is free. So if you wanna take that software and put it on a bunch of your laptops or analysis station or whatever, go for it, that's free.
We actually, we had a bit of a question with that.
Yeah.
So can that be used to replace FlowJo partially? Cause it
Yes.
seems like it can.
Yes.
Yeah, nice.
There are a lot of analysis tools in the Sony softwares that are very nice, you won't find in FlowJo. I also think there are things in FlowJo that FlowJo does better, I'm just gonna be honest. And then there are some labs where like they have their whole, you know, like workspace built out in FlowJo and it's just easier for them to transfer the files. But I have talked to many customers who have essentially stopped using FlowJo.
Anyways,
Or they've stopped using whatever third party they're using, because there's a lot of tools in there, or they use the tools in the Sony to sort of get their data looking the way they want, they re-unmix, they do autofluorescence, they do all that, and then they move it to FlowJo or XDS Express or whatever it is they're using to then do their reports or whatever. Some people do like a combo of the two. Yeah,
So
certainly.
this software is compatible to transfer to transfer to that.
Yes, you can always export XDS files and take them anywhere you want. You can get them unmixed, you can get them raw, you can sort of do, if you do a bunch of adjustments to your mixing and you have 4 unmixing matrices, right, when you export your XDS files, it's gonna say which ones do you want? You want all 4 sets of data? You can choose the one you want, you can do whatever you want. Or you can take what we call XDAT files, re-import them into a Sony software and you have everything. Right, you have your unmixing, you have your worksheet, you don't have to rebuild anything. So that's why some people like going to our software as well, because when you go into a third-party software, you then have to rebuild your gating strategy or whatever it is, right? So there's those things to think about as well. All right.
And use your software, you can generate the report or even publish data or whatever.
That's where I think FlowJo is a little stronger, to be honest with you. You can export PDFs of your workspaces for sure in the Sony software. It is 21 CFR Part 11 compliant. So if you're someone looking to do like, clinical samples or anything like that, it has like electronic signatures, it has audit trails, it has all of that if you need it. But if you are somebody that like has like a specific report that you need to like build and modify, I think maybe FlowJo is a little bit better for that. But that all depends on what it is you're doing and how you want to build it. And I don't want to speak for FlowJo either because I haven't used FlowJo in quite some time, to be honest with you. So I don't know for sure. Right?
Because that machine sounds very great.
Yeah.
Yes, very
It does
great.
sound very great, doesn't it?
But one thing is that if there is one or two features you really want to edit on, what that will be.
Because the I put add on.
So your this already wonderful machine.
This is kind of like looking way towards the future, but I would love some kind of like AI integration. Where I could basically give it a certain parameter and it would just do all the adjustments for me. Something like that, that's just me spitballing, I
I
don't,
can put you on there, penalty sign. You told.
Oh, I know design.
Yeah.
Okay.
yeah, that's another thing, yeah, I would like it to basically, maybe if it would auto generate an SSM. You just click an SSM button and it gives me the SSM of the panel every time, I would love that.
I think
But we have some tools that we can.
But then
help
maybe
you out in terms of
understand.
analysis.
I think it's pretty important to guys who are like with developing study material like
Yeah.
that or this instrument.
Yeah, I agree.
And then once you put that note here, don't have nothing on board that can do this right now, okay? So please keep out the voltage the same and it's not broken.
Yeah, you guys do not have to do this yet, but don't go and play with it yet.
Adjusted. This is a really wonderful thing. I'm looking forward to hunting sometimes if you guys have a demo.
Now everybody.
Sure.
Maybe a movie cleaner, same with the seconds. You have that?
Like a volumetric,
Yeah, like the one.
we don't have that yet.
Yeah.
Collection by volume.
Yeah, but you can't give it a volume, you can't say like take 10 microliters, you kind of like back calculate it, but it's a good idea. Yeah. So I have a couple more things I want to show you guys really quick, some new like brand spanking new things. I mentioned our free, sort of our base user software is free, right? Take as many copies as you want, go for it. We also have this SFA, which is called Spectral Flow Analysis, and this is our cloud-based high parameter analysis platform that was just released not too long ago. So what's the big deal with this? The big deal with this is that with conventional analysis, you have all the manual gating, analysis time increases, the number of colors increases, right? It becomes this big complex monster that nobody really wants to deal with, right? So Spectral Flow allows to write 40 plus parameters, and people always want to look at dimensionality reduction, clustering like FlowSum, or things like that, and there's no really good, at least from my understanding, streamlined way of handling this yet. It's always like you have to go to a third-party software and upload and rebuild and all of that. So Sony sort of, recognized this unmet need, right? The ID launched in 2020, we're generating all this great data. Now we have these users with all these like high parameter panels, and we want something that was easy to adopt, high speed, right? Really high efficient and high speed, and was flexible. Okay. And that's basically what they did. So they took the ID 7000 software, the base software, and they basically made the SFA software look exactly the same. So when you go from ID software to SFA, you don't have to learn any new layouts or GUIs or whatever, it looks just like ID software, all the buttons are in the same place. You basically just like upload your XStat file, there's your data, just the way you left it on the ID, your workspace is still there, undisturbed, all that is the way you left it. But now, you can basically do all these sort of unsupervised clustering algorithms, UMAP, bitsnees, tisnees, flowsums. And one of the big ideas was, let's make it fast, really fast, or faster than at least what's available out there. And so Sony worked with the original developer of flowsum and came up with BL flowsum, batch learning flowsum. Okay, so basically the take home message here is that it operates a lot faster than traditional clustering algorithms because of that batch learning component. Okay, so basically the clustering time and all of that is less than other softwares that are out there. But basically here are your clustering tools, the visualization tools. So once you have your flowsum generated and you have all this data, you can then go to UMAP, you can go to bitsnees, right? You have all this information at your fingertips, and it all plays with your original data, right? So you can go back to your ribbon plots, you can go back to your two-dimensional plots, and you can sort of gate on things and see it reflect in the gates that are there. Okay, so that's sort of the take-home message. This is not free. You want to point that out. This is a subscription-based software, right? I don't want to get everyone too excited. This is a subscription-based software. It is not wildly expensive. You can talk to Justin about pricing if you're interested, but it's something that would basically streamline that high parameter analysis if that's something you're interested in. Right? Question about it?
I would think that like clustering cell types would benefit from the multiple fluorescence parameters that AD7.
Yeah, for sure. And I'm gonna take this one step further. So now we have the ID, now we have these advanced analysis platform. What about sorting? Right, let's say you identify these populations on the ID and you're like, oh, this is awesome, never seen these before, I wanna sort them. Later on this year, we are releasing the FP 7000, shameless plug, okay? This is not yet available, but again, if you're interested, talk to Justin, it should be available later on this year. This is an up to 6 laser spectral sorter, completely, perfectly optical matched to the ID 7000. So if you have an ID, and you have an FP, you run a sample on one, you run it on the other, they're identical. Right? Multiple nozzle sizes, 70, 100, 130, 6-way sorting, sort speeds up to 25,000 events per second with really, really nice purities. I've only seen a little bit of introductory data from Japan, and they basically ran, 25,000 events per second, actively sorting with efficiencies of like 98% or something. It was really crazy. Again, like I said, matched the ID 7000, still has our patented unmixing algorithm, the WLSM, our core finder algorithm, which if you use the SH or the MAs, it's all going to be automated. So you won't have to even really know how to run a sorter to run a sorter, right? You're going to put a tube of beads on there. It'll do all the droplet calibrations and side stream calibrations and drop delays and all that for you, okay? It's going to do forward scatter, side scatter, and I don't know if you noticed, but we're adding in a violet side scatter. So if you might be interested in small particles, this is something to consider, and we will most likely, I don't want to promise anything, but think about things being optically matched, okay? And then in terms of input, right, just like our other sorters, 1.5, 5, 15, 50 ml inputs, and then in terms of collection, 1 through 6-way tubes, and then for plates, anywhere from 6-well plates all the way up to 3 to 4-well plates, single cell sorting, index sorting, basically anything our other sorters can do, it will do it here, but now with the spectral. Okay? And that is officially all I have for you. But if you have any other